Hi everyone, Kitty Clinical here with another video, and today we're going to be continuing the biochem discussion via USMLE Step 1 Thoughts. I'm going to talk about the functional organization of a eukaryotic gene. Effectively, there's an enhancer or silencer gene a little bit distant, but going down south, you'll eventually reach the gene itself, where you have a promoter, the 5' UTR, the open reading frame, and the 3' UTR. And then maybe a little bit more further past, you'll see a silencer as well. So this is known as the coding strand, where the promoter is consisting of a cat box, also like a CAAT, followed by a TATA box, or TATAAA. And then that's when the transcription starts at the exon. Then you get intron, which is starts with a GT and ends with an AG. Then you get an exon, and then you get an intron, which starts with a GT and ends with an AG, and so on and so forth. The last exon will have a polyadenylation signal. Usually this is AATAAA. So this DNA is the coding strand. It gets transcribed to pre-mRNA, where you get the exon, intron, and the last exon with the polyadenylation signal. So you get rid of the cat box, the TATA box, and you end up with this series of exons and introns, which then are the pre-mRNA, which gets spliced into a mature mRNA. And the mature mRNA is where you have basically you get the introns. What happens is you get rid of them, and so you have the AUG star codon, the protein coding region, the stop, and then as we talked about, the AAUAAA, which you would have seen from the pre-mRNA, because remember the T becomes a U when you go from DNA to mRNA. So AATAAA in the DNA coding strand, AAUAAA in pre-mRNA. In the mature mRNA, the AAUAAA is followed by a poly A tail, which is like 6 A's basically. And then that gets translated to protein through translation. So let's talk about the different regulation of gene expression. There's the promoter, which is the site where the RNA polymerase II and the multiple other transcription factors bind to DNA upstream from gene locus. AT-rich upstream sequence with TATA and cat boxes, which differ between eukaryotes and prokaryotes. The promoter mutation commonly results in dramatic decrease in level of gene expression, because obviously it's the one that's promoting the gene. So if that's messed up, then the entire thing should be messed up as well. You have the enhancer, which is a DNA locus where regulatory proteins or activators bind, increasing the expression of a gene on the same chromosome. The enhancers and silencers may be located close to, far from, or even within, such as in the case of an intron, the gene whose expression they regulate. You have silencers. We talked about silencers, where the DNA locus, the regulatory protein, the repressor, binds, decreasing the expression of a gene on the same chromosome. And you've got epigenetics, which is changes made to gene expression, such as heritable mitotically or meiotically, without a change in underlying DNA sequence. So primary mechanisms of epigenetic change include DNA methylation, histone modification, and non-coding RNA. And if you need to touch up on DNA methylation, I would go back to the first part of the biochemistry series. RNA processing, this is mainly for eukaryotes. You've got the 5', which starts with G, lowercase p, lowercase p, lowercase p. It's got that capital G with the three subtext, subscript, PPP. That's your cap. Then you've got the coding, and then it ends with the tail, which is the AAAA with an O, with an H, with an HO attached, or OH attached. So you have the initial transcript called heterogeneous nuclear RNA, or hnRNA. Heterogeneous nuclear RNA is then modified and becomes mRNA. So what happens is you have the capping of the 5' end, or the addition of 7-methylguanosine cap, or co-transcriptional. Polyadenylation of the 3' end, which leads to about 200 A's, or a poly A tail, which is post-transcriptional. And then the next process that occurs is a splicing out of the introns, or post-transcriptional capped tail and splice transcript is also called mRNA. The mRNA is transported out of the nucleus to be translated in the cytosol. mRNA quality control occurs at the cytoplasmic processing bodies, so your P bodies, which contain exonucleases, decapping enzymes, and microRNAs. mRNAs can be degraded or stored in P bodies for future translation. You got the poly-A polymerase, which does not really require a template, it's just polyase. And the AAUAAA, which we talked about, is the polyadenylation cycle. A mutation in this signal leads to early degradation prior to the translation. So that's a whole mouthful of words related to gene expression. If you have any questions about this, please leave a comment down below. I'd be more than happy to explain further. But again, a lot of this is step one stuff. You're not going to really know a lot of this when you practice. It's one of those things where you study it and you memorize it for the exam and you forget about it later on. But it's always nice to go back and just read it again if you're bored. Hope you enjoyed this video. See you in the next one.